Hi, Gen Bio. Dr. Fry here. We are looking at plant diversity this week. And I just noticed that this is our week 8 lab, not our week 7 lab. I'm actually going to break this up into two different videos because it is rather lengthy. So this is going to be part 1. Okay, so plant diversity. We are going to look at a range of plants from their ancestral green algae. So not technically considered a plant by most plant biologists. And then all the way up through our flowering land plants. So if you look at this phylogenetic tree, the different clades of plants are shown here on the right-hand side. Land plants encompass all of them that we're going to look at, but they're broken up into separate groups. So the most closely related to our ancestral green algae are the non-vascular plants, sometimes also called bryophytes. Okay, so these guys don't have vasculature, which means that they don't have specialized cells or tissues organized for the movement of water up from the roots or the movement of food down from the photosynthesizing parts of the plant. So they obviously, if they cannot do that, they have less specialized tissue. They're actually going to have to be a lot smaller for one thing, because they can't move these fluids and sugars far distances. So they're going to be a lot smaller than most of the plants that you would see in your environment. So those are non-vascular plants, and we're going to look at mosses and liverworts as examples of those plants. And then the next clade we're going to look at are the seedless vascular plants. Okay, and those include club mosses, spike mosses, quillworts, although we don't usually have an example of those in lab, ferns, horsetails, and whisk ferns. Okay, so these guys, they do now have vasculature, but they don't have seeds. So like I said, the vasculature is going to enable them to grow taller, for one thing, because now they can transport fluids and sugars throughout the plant. However, like I said, they won't have seeds until we get to our next group, the seed plants. And the seed plants are going to be most of the large plants that you see in the environment. So you're probably familiar with the fact that there are gymnosperms, which are the plants that have what we call naked seeds. They have seeds, but they're not encompassed in a fruit. Okay. And then angiosperms are flowering, fruiting seed plants, which we will get to then as we go through the presentation. So we're going to start with, like I said, the non-vascular plants are bryophytes. So let's move to our next slide. Chara, I think I had mentioned in the previous slide, is actually what we believe to be the closest living relative to land plants. Chara, it looks sort of like this. It's considered to be a green algae that grows in freshwater environment. And the reason that we believe it is the closest living relative to land plants is because of several characteristics that it has in common with land plants. And those are listed here. So there is a complex that is used in plants to produce cellulose, which is a major component of the cell walls in plants. And sometimes it takes on a rosette-shaped appearance, this complex. So circular sort of arrangement, or round arrangement to the complex. And other times it's linear. So in most of our land plants, as well as in Chara, it has this rosette shape to it. So it's a small difference in morphology, but it lends evolutionary biologists to believe that Chara is more closely related to land plants than other types of algae would be. Other things that Chara have in common with land plants are the structure of a flagellated sperm. So as you'll see as we go through the talk, plants often reproduce sexually, and the sperm will have a flagellated tail. And so the structure of that is very similar between land plants and Chara. And then it also has a phragmoplast. The phragmoplast is a structure that provides support and scaffolding. It's made up of microtubules, and this provides support and scaffolding for the formation of the cell plate. When cells are undergoing cell division, there's going to be a plate that forms between the two daughter cells that will ultimately become the cell walls of the two daughter cells. And that is supported by this structure called the phragmoplast. And that's present in chara as well as in land plants, but not present in other algae. And then lastly, peroxisome enzymes are present in chara as well as in land plants. So these will enable oxidative reactions and produce hydrogen peroxide. Not all species have peroxisome enzymes. So again, this is just a similarity that led to the belief that chara is the closest living relative to our land plants developmentally. Okay. So they, one other detail, they're often called stonewort. They look like the pictures shown here. This one is obviously in water. This one is laid out flat. They're called stonewort because if you touch them, they often have a grainy or stone-like texture to them. And this is made from buildups of calcium chloride that are present in the cell walls. So that's commonly used as a name for chara, stonewort. All right, so that will lead us to our discussion of land plants. And I should mention here that there's this concept of alternation of generations, which is present in land plants now, but not in our chara and our charophytes. So alternation of generations is a concept that can be seen in plants where one generation, one sort of part of the plant's life cycle is a diploid generation called a sporophyte, and then another part of the plant's life cycle is a haploid gametophyte generation. And they take turns producing each other throughout the life cycle of the plant. So a sporophyte, the word itself means spore producing. A sporophyte is going to be the diploid generation that produces spores. Now the spores are haploid, so obviously the sporophyte, which is diploid, has to undergo a meiotic division, has to undergo meiosis to produce the spores, okay? And then the spores, which have been produced from the sporophyte generation, divide mitotically to produce the next generation called the gametophyte. So these are still going to be haploid because they formed from the haploid spores, and they will produce gametes. So gametophyte is gamete producing. And as you probably are familiar already, gametes are sperm and egg. And so the male gametophyte will produce the sperm and the female gametophyte will produce the egg. And of course, these are both haploid, the sperm and the egg. And that will then, at the fusion of sperm and egg, restore the diploid number. So now we've got fusion of the gametes, sperm and egg meet, you've got a zygote, develops into an embryo. That's obviously going to be a diploid structure. And so that is going to be develop into our mature sporophyte then, okay? So then the cycle just repeats itself with the next generation. So you should definitely be familiar with the concept of alternation of generations as we go through and look at the different types of plants. And that brings me to this slide that gives a nice overview of which generation, the gametophyte or the sporophyte, is dominant in each of our different clades of land plants. So you'll see here in the first column, there are mosses and other non-vascular plants. That was I'm gonna click back, that was our lowest sort of developmentally, our lowest level of plants. And then the next one are seedless vascular plants, followed by seed plants. I'm going to jump back here. So our non-vascular plants is in the first column. Seedless vascular plants is in the second column. and our seed plants, our most developed, largest, most specialized plants are going to be in the final third column. And then what I'd like to point out with this slide is that the gametophyte generation is the dominant generation. So this is the haploid generation, is the dominant generation in our mosses and non-vascular plants. So the non-vascular plants are what you would see when you look at the plant. The larger structure is going to be the haploid generation. So for instance, in this picture here, this is a moss, and the large green structure here, that's the gametophyte generation. That's the haploid generation of the moss. And then it will give rise to and support the sporophyte generation in mosses. So you'll see here this structure growing up out of the female gametophyte is our sporophyte. It's diploid. So in this category of plants, the sporophyte is dependent on the gametophyte. Okay, it cannot survive without it. Now, if we turn and look at our seedless vascular plants, such as a fern, now we're seeing that the dominant structure, the larger structure of the fern or other seedless vascular plant is the sporophyte generation. So this is the diploid generation. You walking through the woods, going for a hike, you might see ferns growing. That large leafy structure, that is the sporophyte generation. And now we're seeing a reduced, so a smaller, but still independent, so still can support itself, gametophyte generation. So it is usually smaller, closer to the ground, but still can exist independent from the sporophyte. And that's typical of our seedless vascular plants. Okay. Now, if we look at our seed plants, then, so like all of the tall trees that you see, all of the garden vegetables and fruits, all of those plants are now going to be seed plants, and they have a dominant sporophyte generation, just like the seedless vascular plants. But in this case, the gametophyte generation is so small, it's usually microscopic, and it is in this case dependent on the sporophyte for survival. So things have kind of swapped as we've moved up in the evolutionary process, whereas the gametophyte used to be the dominant in our lower plant groups. The gametophyte used to be the dominant, and the sporophyte was actually dependent on the gametophyte for survival. That has now completely reversed as we get to seed plants, where now the sporophyte is dominant and the gametophyte is dependent on the sporophyte for survival and continuation of the species. Okay, so just want to give you an overview of that before we now move on specifically and look at our non-vascular plants. Okay. So like I had said in the previous slide, the non-vascular plants are characterized by an independent, dominant gametophyte generation. And the sporophytes are dependent on the gametophytes for nutrition. And they obviously, as indicated in the name, lack vascular tissue. So again, they don't have the specialized cells that move water and sugars throughout far distances in the plant. So they're going to be a lot smaller and less developed. So they also lack seeds as well. Again, we'll get to the seed plants. So these lack vascular tissue, lack seeds. There are a couple examples shown here. You can see mosses growing here, and this is liverwort, and we'll talk about each of those in the next couple slides. So first, let's talk about liverworts. Liverworts are, as I said, non-vascular, non-seeded plants. They're really kind of unspecialized. So if you look here at this bottom right-hand picture, the most obvious structure associated with the liverwort is this sort of leaf-like structure called the thallus. Okay, that green, flat, leaf-like structure. However, this is the most obvious characteristic of the liverwort. It's the most dominant structure. And it's going to be the gametophyte generation. It's going to be haploid. And it does not, the liverwort does not have any specialized roots, stems, or leaves. It just has little pores that are present in the thallus that aid in gas exchange. And it might develop a small stalk if it is reproducing sexually, but there's no stem, there's no leaves coming off of it. This is really, it's just a flat structure for the most part. Now it can, it's capable of reproducing either sexually or asexually. The most simplistic way is obviously asexual reproduction. And if you look at this bottom picture again here, you'll notice there are these small barrel shapes on top of the thallus. Okay. And that barrel is called a gemma cup. And it's used for asexual reproduction. So rainwater, it's very simplistic, just splashes in and kind of washes the gemma out of the gemma cup. And that will lead to formation of a new gametophyte liverwort. So that is one way that the liverworts can reproduce. They're also able to reproduce sexually, and they do this by means of antheridiaphores and archegoniaphores. So if you look here at this top picture now, you're seeing down below there's the thallus, and then there's this small stalk that leads to two different shapes of structures on top. So this one, for instance, is more of like a flower shaped structure on top of the stalk. And this one's more like finger-like projections kind of hanging down from the stalk. So these two are the male and female gametophyte structures. So when they are undergoing sexual reproduction, we're going to have sperm develop from the male structures and eggs develop from the female structures. And these structures, you'll notice the prefixes are different, but many of the words are actually, the base words are the same. So anything that has this anther prefix is going to be associated with the male structures. So it's going to ultimately produce sperm. So this is the antheridium. It holds the sperm, and it is contained within the antheridiophore, this structure here. Okay. And then this archa prefix is going to refer to the female structures. So archegonium is going to hold the egg, and that's present in the archegoniophore, this structure on top of the stem, the stalk there. Okay. So rainwater, again, is responsible for the movement of sperm to the egg. So the sperm will be released from the antheridium, but rely on rainwater to actually get that sperm to the egg in the female gametophyte. Okay, so when that happens, of course, you can have fertilization of the egg. This will produce a zygote, now a diploid, and then that will lead to production of the sporophyte, which is going to hang down below the archegoniophore. Okay, so down here, coming out here where the egg has been now fertilized, is going to form the sporophyte generation. So you'll notice it's much smaller than the gametophyte, right? It's reliant on the gametophyte in liverworts for survival and continuation of the life cycle. So once that happens, the sporophyte will develop and ultimately release mature spores, which have been formed in the sporophyte through meiosis. And then the spores will be haploid and will lead to production of the mature gametophyte generation. So that's the life cycle there. And I'll just show you in the next slide, there are just some better pictures, real life examples of liverworts sort of enlarged here. That's of course the thallus, the gemma cup. And this is a picture that I took from one of the examples we had in lab. You don't see the actual stalks or archegonia or antheridia present in this example. We may or may not have those available, but if we don't, you can reference the picture here. Okay. All right, so let's move on and talk about mosses. These are still non-vascular, non-seeded, so they're still bryophytes. They do have now the beginnings of some specialized cells for water and food conduction, but they don't have true tissues that we'll look at later. So as we get to the more developed plants, you'll hear the word xylem and phloem. Xylem are going to be cells and structures that transport water far distances within the plant. And then phloem are similar structures, but for the movement of sugars throughout the plant. So mosses are still non-vascular, meaning they don't have those xylem and phloem, but they are beginning to have some cell specialization for conduction of water and food. So hydroids are the cells that can transport water. Leptoids are the cells that can conduct food in our mosses. Okay, so this is one developmental difference between mosses and liverworts. Okay, they also have the beginnings of stomata. Stomata now are taking on the role of gas exchange. So gases need to be exchanged between the plant and the environment for respiration. Stomata are little pores that have been formed by two cells that sort of meet up and allow for a pore to be opened or closed. Okay, so there are two cells that will open or close a pore, which will allow for gas exchange in mosses and in higher plants. Okay. All right, so these guys, mosses, you can see here, are still pretty small. Like I said, there aren't really a lot of vascular cells, so no vascular tissue. So you can't have a lot of length to cover for the plant. Okay, so what you're looking at here in the picture, this is again the gametophyte generation, which is the dominant generation for mosses. And then it is shown here in the top picture supporting the sporophyte generation, which is going to grow right out of one of the gametophyte structures. So these reproduce sexually, like I said, through the sporophyte, which is dependent on the gametophyte. And this happens through, again, production of the antheridia and the archegonia. So there's going to be two different male and female gametophyte structures. So if you see the stalk growing out of one, you can identify right away that that is from a female gametophyte, because we're still using the same mechanism that we talked about with the liverworts. Water is going to allow for the movement of sperm to the egg, which will have to then be fertilized to produce the sporophyte, which is characterized by the stalk, which we call the seta, and a capsule, which is the top part here, and it will ultimately hold spores inside the capsule. After it has developed, and within the capsule, cells undergo meiosis to return to the haploid number. The spores will be held in there until the lid on the capsule, which is called the operculum, is sort of removed, is broken off, and that will release the spores to then allow for production of the gametophyte generation again. Okay. So there are some structures you should be able to look at and tell the names of. You should know the gametophyte is the dominant generation. You should know that the seta is holding up the capsule, and then the top part of that is the operculum. And then we will just flip to the next slide to reiterate the life cycle, which I mostly touched on in the last slide, but we'll start here. You see this is the gametophyte. The female gametophyte would have, if sperm had fertilized egg, the female gametophyte would have the sporophyte growing out of it. And then within the capsule are the spores, which are then released. There are two different types leading to the male and female gametophytes, which then produce the sperm and egg, respectively. Okay, and of course, once they fuse at fertilization, now we're restoring that diploid number. So now it's the sporophyte developing. Okay, haploid is everything along these lines, spore beginning with the spores and continuing through the gametophyte generation. When the sperm and egg join, you restore the diploid number and then produce the sporophyte. Okay. All right, let's move on now and talk about seedless vascular plants. So we're moving up now to the point of divergence on that phylogenetic tree at the beginning of the lecture, where now we have specialized tissue responsible for conducting fluid, water, and sugar throughout the plant. Okay, so that's what the vasculature is. These guys still don't have seeds, but as I mentioned at the beginning, we're now going to see a much larger plant due to the fact that they can transport water and sugar throughout the plant. And now the sporophyte is going to be the dominant generation, and the gametophyte is going to be the reduced generation. Okay. So some examples are club mosses, spike mosses, horsetails, ferns, and whisk ferns. So we're going to look at each of those. Quillworts is another example, but we don't usually look at those in lab. So we'll just look at the 5 that we often see in lab: club mosses, spike mosses, horsetails, ferns, and whisk ferns. And as I had mentioned, the vasculature is enabling the plants to grow a lot bigger than our mosses and our liverworts. Xylem is the specialized tissue for water transportation, and phloem is the specialized tissue for sugar transportation. Okay, here are some pictures of each, but we're going to work through each of those. So first, let's talk about club mosses. Club mosses are seedless vascular plants. The sporophyte is going to be the dominant structure right here, and it will produce a small stem that has these cone-like structures called strobili. Strobilus is singular. Strobili is plural. So these strobili then will have little modified leaves called sporangia, and inside the sporangia are where the spores develop. So again, this is our sporophyte generation will produce spores, and that of course has to happen through meiosis to reduce then the chromosome number. So it's now going to be a haploid once the spores are developed from meiosis, from meiotic division. So once the spores are produced, then they are released to form into gametophytes. And gametophytes are then able to house both the archegonium and the antheridium in club mosses. So what that means is that we have what's called a homosporous organism, meaning that there's only one type of spore. It's not separate male and female. Everything is going to be produced from one spore. So you don't need to differentiate between male and female. I bring this up because in the next slide that we will look at, you're going to start having to pay attention again to the prefixes that I mentioned. Archegonia and antheridia, we had talked about, where archa is the female structure and anther is the male structure. That prefix will indicate the male structure. Similarly, if you have a heterosporous plant, then mega is going to tell you that it's the female structure. So megaspore, and micro is going to tell you that it's the male structure. Okay. So pay attention to those prefixes. For club mosses, they're homosporous, so there's only one type, so you don't need to worry about that next. And you don't have to worry about that yet until we get to spike mosses, okay? So spike mosses are heterosporous. They're very similar to the club mosses in that they are similar in structure, although I think the spike moss has more of like a leafy orientation to its greenery, and the club mosses are more like needly, as you'll see in lab. Similar characteristics. One major difference, as I said, is that the spike mosses are heterosporous. So they still produce the strobili, but now there are strobili that produce megasporangia, that produce megaspores, and then separate microsporangia that contain microspores. So these will be the female and male counterparts, okay? And then they will go on to produce megagametophytes and microgametophytes. Of course, the microsporangia will release microspores, which will develop into the microgametophytes. This is all the male structures. And they will ultimately then, once the microgametophyte develops, matures, it will release sperm. Again, the male structure, the male gamete. And then the megasporangia houses the megaspores, which will then develop into our megagametophyte, the female structure, and produce the egg. Okay. Fertilization happens same way. Rainwater helps to wash the sperm and get the sperm to fertilize the egg, and then the diploid number is restored, and you can have that zygote developed into a mature sporophyte. Okay, and that's again what you're seeing here. All right, let's move on to horsetails. The next 3 slides, we're really just going to identify some morphology associated with each of these different examples from our vascular seedless plants. So horsetails, you're often seeing these growing in really wet environments near, say, a lake, growing around that area. So you've got these long stalks, which have strobili at the top. The stalks have a small enlargement called a node, and that's repeated sort of at various points down the stalk. So here's a node, and it would be, down below. And then the spaces in between the nodes are called internodes. And then around the node itself in whorls are little leaves. So we're starting to see, again, some more specialization, right? You've got leaves now on our plant. And if you were to cut open the stem, you'd be able to look at the vasculature a little bit. So in the middle is a hollow center that allows for gas exchange. That's called the pith. And then surrounding that are vascular bundles. So these are where you're going to have the transport of sugar and water. So the phloem, if you look here, this is one vascular bundle. Here's another, and that is enlarged down below here. I'll try to make it a little bit bigger on the slide for you. So here's a vascular bundle. The phloem is responsible for transportation of sugars. The xylem is responsible for transportation of water. And then you have this carinal canal, which in horsetails is a thicker, elongated canal that helps with the movement of water up through the stem. Okay. These are often also homosporous, so only one type of spore, which is the same as club mosses, but different from spike mosses. All right, and then we'll move on to ferns. Most of these are also homosporous, although they can also be heterosporous. But again, we're going to just look at some of the morphology associated with a fern. You've probably seen these, maybe with or without recognizing what they were, if you're ever taking a hike through the woods. Small leafy structures that are kind of growing out of the ground. Could be really pretty. The leaf itself, this portion here is called a frond, and it's attached to a petiole, which is the stem that kind of attaches that big leaf-like structure. The petiole grows out from what we call the rhizome, which is kind of looks like a root, but it's technically a horizontal stem. It runs horizontal, often underground, sometimes just at the surface of the ground. looks a lot like a root, but there's actually separate roots that can grow out from the rhizome. So the rhizome supports the petiole. Petiole supports this whole structure called the frond. Within the frond, there are sort of smaller leaves. So this is called a pinna. The whole thing is called a pinna. And then if you were to enlarge that even further, there might be pinnules coming off of there. So this one little section is a pinna. And then within the pinna, there are pinnules, little more leaf-like structures there. And then once you get to the point where the frond is present, the stem is now called the rachis instead of the petiole. This is a petiole, but this section here would now be called the rachis, as well as here and here and here and so forth. And then if these are still developing, they often take on an appearance as in the bottom right hand picture here. So there's this like curled up structure coming out of the rhizome, and this is called a fiddlehead, the little curled up structure. As the frond is still developing, it kind of uncurls, and that process is called circinate vernation. It's the uncoiling of the developing frond. Okay, while it's still coiled, it's called a fiddlehead, probably because it looks like the head of a fiddle. All right, and then on the underside, if you look at a mature frond and you turn it over and look at the bottom, you might find little dots like shown here in this bottom left picture. These are called, the little groups of dots are called sori. The sori can have a little covering on top called indusium. And this is where your spores are going to develop, right here in the sori. Okay. So next time you're taking a walk, if you're out in the woods, take a look at the ferns, flip them over, see if you can find the sori, and then you would know that in there is where your sporangia and your spores are going to actually develop. Okay, so again, this is our sporophyte generation. It produces the spores. The spores, which are going to be haploid, are going to produce the gametophyte generation, and still haploid, until they produce gametes, which fuse to restore the diploid number and produce our mature sporophyte, which forms from this uncurling of the frond. Okay. All right, that's it for ferns. Let's move on to whisk ferns. Whisk ferns are another example of vascular plants. They have a stem with no true leaves, but instead some branching, where you're going to see these little bulbs. Little bulbs that are on each of the little branches are actually the sporangia, okay, where the spores are going to be developing. So I have just picture the branch of the whisk fern, and then that's enlarged to show you there are these little sporangia, and then that's enlarged even further to show you spores that are able to be released from the sporangia. Okay, so the sporangia for whisk ferns are often grouped in sets of 2 or 3. This one has 3, 1, 2, and 3 sporangia. And when that ruptures, it releases the spores, which again are enabling the growth of the gametophyte. Okay. They also have rhizomes instead of roots. This allows for water and nutrients to come up from the ground and reach the rest of the plant. Okay, this is where we're going to take a short break because I feel like it's a lot of material, and we're going to end this so that you and I both can take a little break. And then when you have the opportunity, come back and view the rest of this presentation. So we're going to start talking about seed plants in the next section and move on to some details about the roots and stems of angiosperms. So I'm going to stop here for now, and I'll see you in the next video.